Okay, right. Good afternoon, everyone. Okay, jadi there are about 128 of you now. Tinggal 7 orang lagi lah. Okay, anyway, we're just going to continue, okay? Right, so we try to make it secepat yang mungkin lah hari ni. Okay, but then again, there are so many things that we need to cover juga lah. Alright, okay. So let me just recap on what we read ataupun what we did last week. Okay, jadi pada minggu lepas, okay, we went into the first 2 lecture of our class. Okay, boleh dengar kan? Okay, you can hear me?
Boleh, Doktor.
Boleh,
Okay,
Doktor.
Boleh, Doktor.
Alright, okay, thanks. Right, so in the first 2 lecture, okay, kita telah cover pada minggu lepas so we started to talk about metabolism. Alright, so ingat when you were doing biochem in your first year, you are basically looking at the structure. Okay, macam mana bentuk gula, macam mana bentuk glucose, macam mana bentuk fatty acid, how does amino acid look like. Okay, so that was what you covered in your first year. Okay, now in this class, okay, you are going to go beyond that. Okay, you're not just going to know how to draw them. You're not going to know just they are 6 carbon ke, dia ada bentuk macam ni. Okay, but you are going to see how they are used or how they are metabolized. Okay, sama ada macam mana mereka dibentuk ataupun macam mana mereka digunakan. Okay, contohnya kalau kamu makan nasi lah. So that's why we started with the metabolism. Okay, so in this year punya kursus untuk semester ni lah 2043, you'll be looking not just looking at the structure, but you're looking at how the structure is manipulated enzymatically lah dalam badan kamu. Okay, sama ada badan kamu pecahkan untuk menghasilkan tenaga ataupun badan kamu bentuk untuk badan kamu gabung-gabung to produce biomolecules. Okay, so that is what we're going to do this year lah. Okay, so we talk about metabolism. So we talk about what is metabolism, what is we know that there are 3 types of metabolism. Okay, firstly you have anabolism. Okay, anabolism means you are building things. Okay, maksudnya daripada carbon, daripada hydrogen, or from water, from CO2. Okay, you're building katakan monosaccharide. Okay, then from monosaccharide you're going to build disaccharide. From disaccharide you panjangkan dia, then you're going to build polysaccharide. Okay, so that is anabolism. Now catabolism is the other way around. Okay, catabolism is when you are going to break the big biomolecule. Okay, contohnya kalau kamu makan nasi. Okay, you all know that when you makan nasi, basically what you have in your rice is polysaccharide. Okay, and that polysaccharide adalah dalam bentuk starch. Okay, so you have starch in rice, you have starch in mee, okay, you have starch in kuih tiaw, you have starch in, itulah asalkan dia karbohidrat lah. Okay, so it is basically starch. Okay, now some of you still tertanya-tanya juga, how is the starch different from say rice dengan mee, nasi biasa dengan nasi pulut? Okay, they're all starch. Basically, they're all starch. Okay, which basically matter of glucose chain. Okay, glucose dicantum panjang-panjang. Okay, jadi kamu tanya, doktor, macam apa pula beza kalau dia beras wangi dengan beras tak wangi, or beras wangi dengan beras pulut, ataupun beras murah dengan beras wangi, ataupun beras murah dengan beras pulut? Okay, have you ever asked yourself that? Okay, are they the same starch or not? Basically, they are the same starch. Okay, only that when you talk about starch, you know there are 2 types of starch. Okay, the first type of starch is called amylose. Okay, amylose is a long chain of apa namanya glucose. Okay, so dia tak ada branch. It's just long branch. It's just a long chain. Okay, now those long chain you usually find them in beras yang murah. Okay, beras yang 1 Malaysia ke, beras yang harga sekilo RM2. Okay, so you find them in those kind of beras. Okay, and those beras actually they are more readily digested by your body. Okay, sebab dia rantai panjang saja. Okay, and then how is it different from beras wangi? Okay, beras wangi dia juga terdiri pada long chain of glucose, but dia ada branch. So these are amylopectin. Okay, you know you have amylose, which is just long. Amylopectin is the branching. Okay, so kalau beras wangi, you have a lot of branching, banyak branch. Okay, you have the glucose, then you have the branch. Okay, so those are beras wangi. Okay, pulut is even more branchy. Okay, sebab itu kalau kamu masak lah. Okay, if you cook beras murah, okay, normally you put a lot of rice, betul kan? Supaya dia cantik lah, sedap. But when you cook beras wangi ataupun when you cook beras pulut, you kurangkan air, betul kan? Okay, your mother say, letak sikit air when you masak beras wangi. Okay, why? Because it's already branching. Okay, kalau beras murah is very is very long. The molecule is very long. So you need to put more water so that you break the molecule. Okay, you break the molecule of the starch. But when you have beras pulut ataupun beras wangi, what happens is that they are more branchy. So you need less water to go inside. Sebab memang dia dah kembang. Okay, molecularly dia memang sudah kembang. So that's why you need to put less water. Kalau tidak, when you put the same amount of water dengan beras murah, dia jadi lembek. Okay, dia jadi bubur. So that's the reason behind it lah. Okay, so when you have a long chain of polysaccharide, it's the same lah. In mie pun sama, dalam kuih tiaw pun sama. As long as it's starch, okay, it's starch. Only that sometimes the starch has some protein, then you get tepung gandum. Okay, when you have when you have carbohydrate that has no protein, then you're gonna get rice. Okay, sebab itu you buat kuih tiaw. Okay, kuih tiaw dengan mie lain. Okay, mie lain sebab dia banyak protein. Kuih tiaw ataupun mihun is still starch. Okay, but they are different. They are white because they have no protein or they very little protein. That's why bentuk dia macam itu, right? Okay, but eventually when you do eat carbohydrate either from your nasi or your mie, okay, the first line of catabolism is in your mouth lah. Okay, your amylase will start to break into smaller. Mula-mula dia punya chain is very long. It's 1,000, it's 10,000. Okay, but once you start to kunyah, dia akan jadi pendek sikit lah, mungkin around beberapa ribu. But your amylase in your mouth will start to break. So, they can break menjadi oligosaccharide. Okay, so you become oligosaccharide. And usually, the longer the chain, the less sweet it is. Sebab itu kalau kamu makan beras, beras tak manis. Okay, but when you eat sugar, sugar is a disaccharide. Tahu kan? That's why dia manis. Okay, it reacts differently in your mouth. Yeah, all this you belajar lah. These are all biochemistry stuff. Okay, so your amylase will start to digest the beras, the starch into shorter chain. Okay, and as the shorter or the oligosaccharide goes into your stomach, into your usus, okay, it is further digested. Okay, so daripada beribu-ribu dia menjadi berpuluh-puluh. Daripada berpuluh-puluh dia akan jadi beratus-ratus. Daripada beratus-ratus dia akan jadi double unit. Okay, so double unit is the disaccharide. Okay, so the disaccharide that is formed from your starch is called maltose. Okay, kalau sucrose adalah gula. Gula is sucrose, which is basically fructose plus glucose. Okay, kalau starch, when you break or when the enzyme breaks menjadi dua, okay, become disaccharide. This disaccharide is called maltose. Okay, basically it's an alpha glucose. Okay, so the alpha glucose that is broken and become glucose. Okay, monomer lah. Say there's the C6. So this C6 that your darah bring into your different cell to be firstly dia akan masuk dalam proses yang pertama ni, glycolysis. Okay, so glycolysis is itulah when your body starts to receive glucose, then start to break the 6 carbon of glucose into 3 carbon of pyruvate. Okay, so we covered that. Okay, so glycolysis maksudnya kalau kamu baru makan tengah hari tadi, if you makan pukul 1, pukul 12, probably you are just entering glycolysis lah. Okay, you dah cerna dekat mulut, cerna dekat usus, it's absorbed into your blood. Okay, it's absorbed into your cell. Where inside your cell you start to break it into pyruvate. Okay, so that was glycolysis. Okay, so what happens after glycolysis? Okay, now we all know glycolysis. The last part of glycolysis is pyruvate. Okay, so the last part is pyruvate. Jadi itu yang kita akan masuk sekarang. Okay, what happens to the pyruvate? Okay, now glycolysis happens in your cytoplasm. Okay, if this is your cell, if the glucose is absorbed inside, okay, so in the cytoplasm they start to be broken into pyruvate. So that happens in the cytoplasma to produce pyruvate. Okay, so what happens to the pyruvate? Okay, the pyruvate will enter your mitochondria. Okay, in the mitochondria matrix, disitulah dia akan mula it will be digested again, or it will be catabolized again into acetyl-CoA. Then it will enter the Krebs cycle. Okay, so Krebs cycle ataupun the TCA cycle is the same thing. Okay, jadi kita akan masuk dalam TCA cycle. Okay, assuming you dah ada glucose, your glucose has been converted into pyruvate in your cytoplasm. Okay, from your cytoplasm, it's brought into the matrix of your mitochondria. Okay, then TCA ataupun Krebs cycle will start to happen. Okay, alright. Jadi kita akan masuk dalam lecture ketiga, then after that we're going to go into lecture keempat. Okay, so now we're talking about what happens to the pyruvate. Okay, what happens to the pyruvate from your gula. Okay, which is in the mitochondria matrix. Okay, boleh? Alright, so we're going to go into TCA cycle. Okay, now TCA cycle, dia ada banyak nama. Okay, some book call it TCA cycle, the tricarboxylic cycle. Some call it the citric acid cycle. Okay, some book call it the Krebs cycle. Okay, Nama lain-lain tapi benda yang sama. It's the same reaction. Okay, for me, I always call it Krebs cycle. Okay, because it's very short. Okay, lagi senang nak sebut daripada saya nak sebut TCA, daripada saya nak sebut tricarboxylic. Okay, menyusahkan lah. So I just say Krebs cycle, Krebs cycle. But it's the same thing. Okay, so if you're familiar with the Krebs cycle, and I'm sure you pernah dengar, then it's the same process lah. Alright, so we're going to go into Krebs cycle today. Okay. Let's change. Okay, so we're going to go into lecture 3, which is the Krebs cycle. Okay. Now, the Krebs cycle, okay, the TCA cycle, banyak nama lah. Okay, citric acid cycle, Krebs cycle, it's the same thing. Okay. Now, so basically, Krebs cycle happens again in the matrix of the mitochondria. Okay. It happens, mula-mula dia akan terima pyruvate dulu. It's the same pyruvate from the glucose that you makan. Okay. But before pyruvate is being used, sebenarnya pyruvate is further degraded. Okay, dia tak terus ambil pyruvate. Okay, pyruvate memang dia ambil, but it will degrade into, pyruvate is 3 carbon. Okay, what happens is that it will degrade, it will take 1 carbon untuk tinggal 2. Okay, it becomes acetyl-CoA. Okay, 2 carbon. It's a 2 carbon, right? So during a TCA cycle or Krebs cycle, you will produce more ATP because that is what you want. Okay, you want to produce ATP for your energy. Okay, ingat lah, kalau your glycolysis, how many ATP? You're producing 2. Net lah, net. Okay, actually you produce 4, but 2 you guna, okay, in the first part of the process. So during your glycolysis, you produce 2 ATP, and then during the TCA, you produce some more, but actually what is more being produced during the Krebs cycle is the NADH. Okay, NADH ni yang banyak dihasilkan. And this NADH will go to the matrix, not to go to the internal membrane of your mitochondria to produce more ATP. Okay, but just know that you produce more ATP and you produce a lot more NADH during the Krebs cycle, right? Okay, so this is the Krebs cycle yang kamu belajar pada first year dulu. Okay, now from this Krebs cycle, you have to know what happens during the cycle itself. Okay, remember, if this is your, katakan this is your matrix, the matrix of mitochondria, so you will receive the pyruvate. Okay, pyruvate from your cytoplasma will be transported into your mitochondria. Okay, so what happens in the mitochondria? So this pyruvate will undergo an initial step. Okay, this is the initial step. Okay, where you have the acetyl, the CoA enzyme. Okay, and acetyl, the enzyme. Okay, and then you have NAD+. Okay, to react with pyruvate. Okay, pyruvate berapa carbon tadi? Pyruvate 3 carbon. Okay, ingat lah, 6 carbon of glucose dapat 2 pyruvate. Okay, pyruvate 3 carbon. Okay, so what happens when pyruvate comes in? The first thing is that it will take one of the carbon, kan dia 3 carbon. It will take one of the carbon away. Okay, so pergi mana carbon dia? That carbon will become carbon dioxide, CO2. Okay, so from 3 carbon dia akan jadi 2 carbon. Okay, satu ni dia akan jadi CO2. Right? Okay, so 3 carbon, hilang 1 carbon in the form of CO2, okay, producing NADH, okay, and what happens is that 3 carbon akan menjadi 2 carbon, okay, so acetyl-CoA is actually 2 carbon, okay, so 3 carbon here, 2 carbon here, alright, so acetyl-CoA 2 carbon, right, and then as it enters the citric acid cycle, dia akan bergabung dengan oxaloacetate, okay, oxaloacetate is a 4 carbon. Okay, 4 campur 2 dapat 6. Okay, you get 6. Okay, so you get citrate. Okay, so the first metabolite you get in the citric or in the Krebs cycle is citrate. Okay, citrate is 6 carbon. Okay, so citrate, okay, in your first year, this is your first year. Okay, citrate in the first year dia akan terus pergi membentuk isocitrate. Okay, now from citrate to isocitrate, there is just a change of molecule. Nothing is taken away, nothing is added. Okay, so basically ini adalah 6 carbon, ini adalah 6 carbon. Okay, sebab itu dia nampak kuning sini. This is yellow sajalah. Okay, color coded for you. Okay, 6 carbon, 6 carbon. It's just rearranged. Dia rearranged, but still 6 carbon. Right? So between isocitrate and then between after isocitrate is produced, okay, then the next thing in the Krebs cycle is the production of alpha ketoglutarate. Okay, so this is 5 carbon. Okay, alpha ketoglutarate is, this is 6 carbon. Alpha ketoglutarate is 5 carbon. Okay, so daripada 6 okay, carbon menjadi 5 carbon. So sebab kamu kehilangan carbon, so where does the carbon goes to? Ni, dia mengeluarkan CO2. Okay, so this is carbon dioxide is reduced here, is produced here. It is also at this point you're also producing NADH. Sama juga, produce CO2 here and NADH. Sama juga, produce CO2 here and NADH. Okay, basically this dia ulang balik. Okay, so you keluarkan CO2. So 6 carbon, 6 carbon, 5 carbon. Right? So from 5 carbon, alpha ketoglutarate, you in the Krebs cycle, it will then produce succinyl-CoA. Now, succinyl-CoA is 4 carbon. So basically, you have 6 carbon, 6 carbon, 5 carbon, hilang 1 carbon dioxide. From 5 here, hilang lagi 1 carbon, okay, sama. You lose 1 carbon dioxide and you get NADH. Then this one becomes 4 carbon. Okay, 6, 6, 5, 4. Everything else becomes 4. Sebab itu dia letak sini kuning, kuning, or orange, orange, green, everything else is blue. Okay, so when you look at Krebs cycle, okay, the first thing that you have to know what happened is that the number of carbon. Okay, so 6 carbon hilang 1 jadi 5. Right? 5 hilang 1 menjadi 4. Okay, and then along this process there is no more loss. Okay, 6, 6, 5, 4, 4, 4, 4, 4. Okay, so that is where now when your brother ka, your sister ka, or your uncle ka, your auntie or your friends ask you, oh, where do you get the carbon dioxide from? Jadi macam mana kamu nak jawab? Then they say, oh, from your liver, from your liver, from your lung, okay, from your blood. Yeah, but where is it from? Like like you bernafas, respirasi. Yes, but where it's from? Okay, from there. You tak dapat jawab. Okay, now you know how to jawab. Now you know that actually the CO2 that you're producing, a lot of it is actually from the Krebs cycle, right? So you get it here, CO2 menghasilkan CO2 sini. From your between isocitrate dengan ketoglutarate, you get CO2. Okay, between this and this, you also get CO2. Okay, so lain kali kalau orang tanya kamu daripada mana datang CO2, it's from the Krebs cycle. Okay, jangan kamu dari sana, sini situ mana, daripada nenek ke apa, bukan Okay, daripada paru-paru it comes from the breeding. Memang daripada breeding lah, okay. But actually where it's from, it's actually from your Krebs cycle. Okay, jadi sekarang kamu boleh jawab lah, wah pandai. Daripada mana datang CO2? Pada Krebs cycle. Okay, because you're losing carbon, right? Okay, so now in the first year, siapa yang ambil kelas saya? Okay, yang pernah ambil kelas saya ada ajar dia macam mana nak ingat ni, because you have 7 to 8 component here. Citrate, isocitrate, alpha ketoglutarate. You have to remember all these metabolite. Okay, citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinat, fumarat, malat, oxaloacetate. Okay, jadi saya punya cara kotor sikit lah. Okay, but I tell my student and I tell myself lah, so banyak yang kamu kena ingat. Okay, I remember like this. Cikgu itu kata saya suka mak orang. Okay, fumarat. Okay, fumarat mak orang. Okay, so that is how you remember. Okay, you have to remember this because you have to know all the different metabolite. Okay, that is first year. Okay, that is first year. Okay, now in your second year, you have to not only do you have to remember all these different metabolite. Okay, each of these you call metabolite lah. Metabolite 1, metabolite 2. Okay, but you have to know again when we started class last week. Okay, I was saying you have to know what happens to the molecule. Sama ada kehilangan carbon, menambah carbon, kehilangan air. Okay, lepas tu you have to know what is the enzyme involved, and then you have to know what is the peristiwa yang berlaku. Jadi selain itulah, kalau first year kamu setakat ingat saja, okay, cikgu itu kata saya suka mak orang, okay, you just remember that. But at your point now, at your level now, you don't just know that. Okay, you have to know what happens to the individual metabolite. Okay, and in fact, kalau if you're in the first year, you remember this 1, 2, 3, 4, 5, 6, 7, 8. Okay, but actual fact sebenarnya dekat tengah-tengah ni there is another intermediate. Okay, sebenarnya in between isocitrate and alpha ketoglutarate there is also another intermediate which you will have to know this year. Okay, so kamu dah tahu 8, sekarang kamu kena tahu 9 atau 10 metabolite. Okay, so this gambar aja that I'm showing you is just to show the number of carbon. Okay, macam pyruvic acid or pyruvate, 3 carbon, 3. Lepas tu dia kehilangan 1 CO2, menghasilkan juga NADH in the process. So you get 2 carbon of acetyl CoA. So 2 acetyl CoA tambah 4 oxaloacetate, jadi dia jadi 6 citric acid. So just to show lah ada 6 sini. Okay, so that is why the Krebs cycle is actually amphibolic. Amphibolic maksudnya dia ada penambahan, dia ada pemecahan. Penambahan dia berlaku dekat sini sajalah. Okay, lepas penambahan ini, everything else dia pecah jadi 3, pecah jadi 5, pecah jadi 4 carbon. Alright, so this is the Krebs cycle. Alright, now you have to know there is another one sini. Okay, kita akan masukkan nanti. Alright, so does that somehow refresh your memory in your first year? Okay, sepatutnya for Krebs cycle, what you take from your first year Krebs cycle, you should at least know what happened. Okay, what are the metabolite and kemana dia punya carbon itu hilang. Okay, basically it's just the losing of the carbon. Okay, you lose 1 carbon here from pyruvate jadi 2. Okay, you lose another carbon here, and then you lose another carbon here, and alongside you produce NADH. Okay, you produce NADH, you produce NADH, you produce NADH. Okay, at one point you're also producing FADH dekat sini. Okay, and then you're also producing 1 ATP. Okay, dekat sini dia guna GDP lah. I will tell you what's the difference later. Okay, so basically during the Krebs cycle, by the end of the Krebs cycle, you produce some ATP dekat sini. Okay, you produce a lot of NADH, you produce dekat sini, you produce dekat sini, you produce over here, and you produce over here. Okay, that will be your main product. Okay, but what happened? You're actually losing carbon, especially in this area lah, menjadi CO2, which you breathe out, or your cells, many of the time your cell you will reuse lah somewhere dia akan guna balik. Alright, so that is the mukadimah. Okay, that is the mukadimah, or that is the introduction to TCA. Okay, now let us look at TCA in more detail. What happened to each of this one metabolite? Okay, what are the enzyme involved? Okay, right. So kalau tadi from citrate membentuk isocitrate. Okay, now this is your level. Okay, so between citrate, okay, actually between citrate and isocitrate there is actually another intermediate. Okay, this is called cis-econitate. Okay, just in addition to this one, and actually there is an addition here dekat sini ya. Between isocitrate and alpha ketoglutarate, actually there is an intermediate here, and actually between alpha ketoglutarate and succinyl there is another intermediate. Okay, but that kita tak cerita lah. Okay, that one cerita lain kali ya. Okay, I think by the time you nak masuk cis isonitrate, isonitate, you pening kepala sudah. Right? Okay, so just know okay, what are the intermediate? Okay, what happens to the carbon? Where do you lose the carbon? Okay, when is NADH and is produced? Okay. Alright, so this is citric acid cycle or TCA. Okay, so I go on yang kamu kena extra lah untuk hari ini, okay, for this kursus. Right, so remember in the first part of the class I told you lepas kamu makan the 6 carbon sugar, okay, will be produced, will be converted into pyruvate, okay, jadi pecah 2 lah pyruvate. Okay, so this happens in the cytoplasm. Okay, so pyruvate is then, okay, that pyruvate that is produced in the cytoplasm. Okay, the pyruvate is translocated, okay, ataupun dipindahkan. Okay, is translocated from the cytosol or the cytoplasm, okay, into the mitochondrial matrix. Okay, so dia akan masuk dalam mitochondrial matrix. Okay, dia akan guna satu gate. Okay, this gate is known as pyruvate translocase. Okay, so bentuk dia macam ni. Okay, there is a gate here. So it's a protein, it's a transmembrane protein. Okay, that this protein, okay, is situated on the membrane of the mitochondria. Okay, kalau kita tengok, this is cytosol. Cytosol is this part lah, which is your mitochondria. Okay, which is your cytoplasm. Okay, and this is your mitochondria. Okay, you have the outer membrane, you have the inner membrane. Okay, so the pyruvate that is in the cytosol from your glucose tadi, okay, it is brought into the matrix, okay, by an enzyme that is called pyruvate translocase. Okay, so dia akan bawa masuk. Okay, so pyruvate dekat luar bawa jadi pyruvate masuk. Okay, alright. So before pyruvate enters the citric acid cycle, dia akan mula-mula converted into acetyl CoA. Okay, ingatlah from 3 carbon of pyruvate, take 1 becomes CO2. Okay, tinggal 2. Okay, which is your acetyl CoA. Okay, so pyruvate ini, okay, akan bertindak balas. Okay, take 1 carbon away from it, then dia jadi 2 carbon, which is acetyl CoA. So this is the coenzyme A. Dia akan gabung dekat sini. one of the carbon, dia akan dikeluarkan menjadi CO2. Okay, alright. So pyruvate is oxidatively decarboxylated, maksudnya dikeluarkan satu carbon lah. Decarbosylate maksudnya kamu keluarkan carbon. Okay, so satu carbon dikeluarkan secara oxidatif, meaning add oxygen. Okay, sebab itu mengeluarkan menjadi CO2. Right, so the 3 carbon of pyruvate ambil satu, okay, is added oxygen, okay, menjadi CO2. Okay, right? Then you produce 2 carbon, okay, to form acetyl-CoA. Okay, now this enzyme is called pyruvate dehydrogenase complex, PDC. Okay, it is a very complex enzyme. Okay, in fact, it is made out of three different enzymes. Okay, right? So just know that bawa masuk, okay, bawa masuk dalam the mitochondrial matrix and then convert into acetyl-CoA by PDC. Okay. Right, now PDC is a very complex enzyme. Okay, it is actually an enzyme, dia macam ada 3 unit. Okay, 3 unit. Okay, so it is composed of 3 enzyme. Okay, now the first unit E1, okay, the first subunit is called pyruvate dehydrogenase. Okay, dan dia memerlukan TPP. Okay, TPP is thiamine pyrophosphate. Okay, I will explain later. Okay, so just know that pyruvate converted into acetyl-CoA memerlukan PDC, and PDC is made of 3 different enzymes. Okay, enzyme number 1 is called pyruvate dehydrogenase. Okay, and then after that, enzyme number 2, still in the same complex, okay, is called enzyme number 2 is called dehydrolipoamide acetyltransferase. Okay, so cofactor dia adalah lipoamide dan CoA. Then the third enzyme is dihydrolipoamide dehydrogenase. Okay, cofactor dia adalah FAD NADH. Okay, jadi kalau kamu tengok ini, peningnya kepala. Okay, now let me break it down to you. Okay, now cofactor is basically an extension of the enzyme. Okay, without a cofactor, the enzyme will not work. Okay, macam kalau kamu ada pisau lah. Kalau kamu ada pisau, kalau kamu tidak ada punya bilah itu, the pisau will not be able to cut. Okay, all you have will be the hulu. Okay, will be the handle. Okay, so for the first enzyme, the cofactor is TPP. Now TPP stands for thiamine pyrophosphate. Okay, these are TPP. T stands for thiamine, P is pyro, okay, P is phosphate. Okay, so thiamine pyrophosphate, TPP. Okay, now without TPP, the first reaction will not happen. Okay, so TPP is very important. Now again, if you ask me, doktor, apakah itu TPP? Very easy. Okay, thiamine ni apa? Thiamine is vitamin B1. Okay, if you don't have enough vitamin B, okay, you will not be able to effectively use pyruvate ataupun convert pyruvate. Okay, so thiamine is vitamin B1. Okay, now what does it mean by pyrophosphate? Now pyrophosphate maksudnya 2. So you have vitamin B1 tambah dengan 2 fosfat. Jadi kadang-kadang diphosphate, you know, sometimes you use diphosphate, biphosphate. Yang ini dia guna pyrophosphate. Okay, so 2. Okay, maksudnya 2. Okay, you know, when you eat vitamin B, okay, sebenarnya the vitamin you makan tu, if you eat as is, dia tak ada, it doesn't do much. Okay, it has to be converted. In this case, thiamine, vitamin B1, you have to put into phosphate. Then it will work into this one. Okay, so thiamine pyrophosphate, vitamin B1. Okay, now let's look at vitamin E3 pula. Okay, vitamin E3, okay, kamu nampak ni cofactor. Cofactor is FAD. Okay, flavin adeno something lah. Okay, FAD lah. Okay, so this is responsible untuk enzyme yang ketiga. Okay, what is also needed, the cofactor is NAD+. Okay, without FA, without FAD, without NAD, this will not happen. Okay, yang ketiga ni tak akan berlaku. Right? Now again, what is FAD and what is NADH? Now FAD is flavin adeno. Okay, now what is flavin adeno? Okay, it is vitamin B2. Okay, and then NADH is nicotinamide adenine. Okay, nicotinamide adenine, what is it? It's vitamin B3. Right? So this just to show how much important is vitamin B to your metabolism when it comes to TCS cycle, Krebs cycle. Okay, sebab itu sometimes if you are lethargic lah, kamu penat, lepas tu You go and see the doctor. Kenapa kamu penat? Cukup tidur kah? Cukup. Cukup makan kah? Cukup. Are you stressed? Mungkin lah. Tu semua. Tu one of the things the doctor will provide you is vitamin B complex. Betul kan? Some of you, if you're tired, you say that rehat cukup, ini pun cukup, itu pun cukup. Probably what you lack is vitamin B. Okay, so that's why the doctor prescribe to you vitamin B complex. Why? Because B complex dia sangat penting. Okay, without vitamin B1, you don't have pyrophosphate for this one. Okay, without vitamin B2, you cannot have flavin. Okay, without vitamin B3, someone say, kalau tak ambil B complex, betul lah. Because if you don't take, meaning your body cannot utilize the pure fat. Okay, jadi maksudnya no Krebs cycle lah. If no Krebs cycle, then there is no ATP production. No Krebs cycle, no NADH. Okay, and that NADH, okay, yang kamu hasilkan ini is actually this one lah. Okay, and sometimes itulah because you lack of vitamin B3. So itu you get very tired. Okay, not because you tak cukup makan, not because you tak cukup rehat, okay, not because you are sick, okay, but simply because you tak cukup vitamin. Okay, so that's why I will always tell the student kalau dapat duit PTPTN, okay, jangan beli handphone, jangan beli data, jangan beli tudung semua. Okay, the first thing you buy multivitamin, especially when it's going to be exam. Okay, because you can never have enough of multivitamin. Okay, this is one good example, right? Okay, even though some doktor, ramai doktor ada cakap tak payah makan multivitamin lah, makan makanan seimbang. Masalahnya student mana seimbang? Okay, kalau tak ada duit makan Maggi, tengah hari breakfast Maggi, tengah hari Maggi, malam Maggi. Okay, there's no vitamin in your body, kan? Okay, so supplement yourself with multivitamin lah, especially kalau nak dekat exam. Okay, sama jugalah to your parents if you're going to dapat kerja nanti kan. Okay, janganlah bagi dia inilah janganlah bawa dia pergi Korea ke mana ke sebagainya. Kalau boleh bagi dia vitamin dulu. Okay, make them healthy first, right? Okay, anyway. Okay, so pyruvate. Okay, so pyruvate before it goes into the Krebs cycle, it's converted into acetyl-CoA. Okay, and how is it being converted into acetyl-CoA? By PDC. Okay, so the first enzyme complex is you must have TPP, vitamin B1. Okay, then the second enzyme complex, okay, this is where you need the E2. Okay, lastly, to produce NADH, okay, is the third enzyme complex, okay, which is the E3. Okay, jadi ini saya dah cerita dengan panjang tadi lah. Alright, so from here, 3 carbon of pyruvate, okay, 1, 2, 3 carbon alright. So 3 carbon of pyruvate will release 1 CO2. This CO2 dia akan keluarkan menjadi CO2 lah, the gas that you breathe out, or your body can use sometime later lah, right? So from here, then it will undergo the first enzyme complex, the second enzyme complex, where it will eventually become acetyl-CoA dekat sini. Okay, acetyl-CoA dihasilkan, and at one part of the pathway, NADH akan dihasilkan. Alright, so the main product that we're interested now is the acetyl-CoA. Okay, so acetyl-CoA is basically after the second enzyme lah. Cuma E3 ni diperlukan jugalah, but to produce the NADH. Right, so from glucose, you makan glucose, you get pyruvate. Okay, pyruvate transported into your mitochondrial matrix. Okay, then it is converted from 3 carbon tadi, then menjadi 2 carbon. Okay, 1 becomes CO2. Okay, so I hope you know that is how your body works. Okay, senang kamu nak cerita nantilah kalau orang banyak soalan dengan kamu lah. Okay, tapi kalau kamu tak dapat jawab, jangan cakap ambil kelas saya. Okay, right. Okay, so the first thing is once you have the acetyl-CoA, baru acetyl-CoA bergabung dengan oxaloacetate. Okay, 2 tambah 4 menjadi 6. Okay, right. So the acetyl-CoA, which is 2 carbon, okay, will enter the Krebs cycle through the citrate synthase reaction. Okay, so this is the only step in the Krebs cycle that involves pemanjangan ataupun bonding. Okay, because otherwise dia menjadi semakin pendek. Okay, only this part dia menjadi panjang. Maksudnya 2 campur 4 jadi 6. Okay, so this is the only step where carbon to carbon bond is produced. Okay, so you have your acetyl-CoA, 2 carbon. Okay, bergabung dengan oxaloacetate, which is 6 carbon. 4 carbon. 1, 2, 3, 4. Okay, so 4 tambah 2. Okay, then you get the intermediate. Okay, I will not talk about intermediate. Okay, I'll just talk straight about the citrate. Okay, ni nanti pening kepala kamu. Okay, so acetal CoA campur oxaloacetate terus bergabung, okay, menghasilkan citrate, okay, which is 6 carbon. Okay, so 1, 2, 3, 4, 5, 6. So that is the first intermediate in the Krebs cycle. Okay, jadi kita tengok balik. Okay, so you have the acetal CoA. Okay, combines with the oxaloacetate, okay, 2 carbon combined with 4 carbon, you get 6 carbon citrate. Okay, so that's the entry. Okay, so that's the first step in the Krebs cycle. Okay, and this first step is catalyzed by citrate synthase. Citrate synthase. Okay, right? Okay. Get citrate. Okay, right. So the combination or the gabungan berlaku dekat sini lah. Right? Okay. Now, so know this reaction, okay, know the enzyme involved. So, itu yang akan ditanyakan kepada kamu. Okay, contohnya, I can ask you in the exam, one of the question that can be asked based on this one, even based on the transition from pyruvate to acetal CoA. Soalan boleh tanya kamu. Okay, in the exam you can be asked like this. Okay, describe in detail the incidence or the mechanism for the conversion of pyruvate to acetal CoA. You have to cakap lah. Then you have to mention all these 3 enzymes. You have to mention what happens. Okay, this gamma raja. Okay, we have to mention what is being produced, what is being reduced. Okay, CO2. Okay, just from this. Okay, and that will give you 12 marks. Boleh jawab tak? Okay, so that can be one question just from this slide. To give, we can ask you to get 12 marks. If you don't answer, if you don't understand me now, and you cannot muntah balik this diagram or this mechanism, 12 markah hilang. Okay, that's why you have to tahulah, okay, all the intricate details. All right. So just example lah of the exam question that we can ask from you. Right. So after citrate, okay, before the formation of isocitrate, okay, for your level kita naikkan satu lagi. Okay, we add one metabolite which is produced by iconitase. Okay, so this is between the isomerization of citrate to isocitrate. Okay, right? So what happened is that so between citrate and isocitrate, okay, there is actually an intermediate. Okay, now this intermediate, okay, dihasilkan, okay, is produced, okay, firstly because citrate is a very poor substrate for oxidation. Okay, maksudnya it has to be restructured. Okay, so what happens is that iconitase isomerizes the citrate to yield isocitrate, okay, which has a secondary OH. So basically from citrate to isocitrate, okay, you are going to add OH. Dekat sini. Okay, so you don't have an OH here. So basically what happens is that during this process, an extra OH is added. Okay, you're adding more oxygen. Okay, because citrate itself is very poor because oxygen there molecularly is not enough. Okay, so you have to add more oxygen to produce isocitrate. Right? So what is isocitrate? Okay, so iconitase. Now iconitase is an iron sulfur cluster, okay, enzyme. Okay, and it will mediate. Okay, it will mediate this reaction. Okay, so basically just to add the OH to the citrate. Okay, right? Okay, so next, okay, from isocitrate to alpha ketoglutarate, okay, you will have oxidative decarboxylation of isocitrate, okay, to produce alpha ketoglutarate. Okay, so basically between isocitrate to alpha ketoglutarate, okay, seperti yang saya katakan tadi lah, there is also actually an intermediate. Okay, cuma kita tak cerita lah. Okay, just know that there is an intermediate. Okay, and between isocitrate and alpha ketoglutarate, okay, NADH is produced, okay, and CO2 is being released. Okay, now basically kalau kamu tengok, if you look at this gamma raja, it looks very simple, but actually this process is the same if you're going to look at this one. Okay, you have CO2 produced, you have NADH produced. Okay, and it's the same here, sini. Okay, sini, okay, you have CO2 produced, NADH produced. It's the same thing. Okay, and actually it gets more complicated lah. Cuma itulah it's just a bit too hard. Kejap ah. Okay, someone coming ah. Okay, alright. Okay, mana tadi? Okay, right, sorry. Okay, so yeah, between isocitrate and alpha ketoglutarate. Okay, right. So in this process, okay, what happen is that you will have the production of NADH. Okay, you are basically having okay, hydride removal. Okay, maksudnya hydrogen is removed from the isocitrate, and after that you have CO2 produced. Okay, dia tak cerita sini lah. Okay, but you know that you just have to know that CO2 is produced. Okay, and from the 6 carbon of isocitrate, okay, you get a 5 carbon of alpha ketoglutarate. Okay, now this reaction dia tak boleh pergi ke belakang. It's not like the first process is now. Between citrate and isocitrate, it's reversible. Okay, kalau kamu tengok you can look at this one see, it's reversible. Okay, but between isocitrate and alpha ketoglutarate is not reversible. Okay, right? So the reaction is metabolically irreversible. Okay? So what you have to know is that NADH is produced, CO2 is also produced at this point. Okay, so between alpha ketoglutarate and succinyl CoA, okay, the enzyme that is involved is alpha ketoglutarate dehydrogenase. Okay, now basically this enzyme take away hydrogen also. Okay, it takes away hydrogen. Usually dia akan pack dekat NADH. Dekat NAD+. So NAD+ take one hydrogen, they become NADH. Okay, and selalunya this is coupled by the losing of CO2. Selalunya lah, not all the time, but many of the times. Okay, jadi kamu tengoklah. So between alpha ketoglutarate, okay, to produce succinyl CoA, okay, you will lose, again, like I say, you lose the CO2, okay, while at the same time you still produce NADH, okay, because one of the hash is released from here to pack on NAD+, so you get NADH, okay. So this is a second oxidative decarbosylation, okay, followed by the first one tadi is previous one lah, okay. So there are 5 enzyme used at this point, okay, TPP, CoA, lipoic acid, NAD+, FADH, which is basically sama dengan ini tadi. This one. Okay, which is yang sama dengan pyruvate dengan ni lah, pyruvate producing the acetyl CoA. Okay, basically ni I go to this one lah. Okay, ni one sama, ini sama, ini sama. Okay, see this and this sama. Okay, so loose carbon dalam bentuk CO2, loose H, okay, and is accepted by the NAD+ to produce NADH, right? Okay. So that is that. Okay, so all the coenzyme is the same. Okay, TPP, your vitamin B1. Okay, NAD+, your vitamin B3. FAD is vitamin B2. Right? Okay. Now between succinyl CoA, okay, between succinyl CoA and the production of succinate, okay, what happened is that there is one event that is known as substrate level phosphorylation. Okay, so in this event, what happened is that GDP, okay, akan menerima satu fosfat from your blood lah, from the surrounding, okay, to produce GTP. Now for this happens in human being. Okay, GTP. Now a nucleoside triphosphate is made. Okay, now this is guanine triphosphate. Okay, in animal, in human being, we produce GTP. Okay, at this point. Okay, but in plant and in bacteria, they produce ATP. Okay, jadi balik kepada our Krebs cycle ni tadi. If you look at this Krebs cycle, now, so between succinyl CoA to produce succinate. Okay, now this shows that GTP is produced. Okay, now if you look at some textbook, dia letak sini ATP. Okay, yang mana betul, yang mana salah, dua-duanya adalah betul. Okay, Only that GTP happens for animal and human. Okay, but for plant and bacteria, at this point they don't produce GTP, they produce ATP. Okay, but if you ask, oh doctor, what is the difference between ATP and GTP? Um, dari segi tenaga dia tak jauh beza. Okay, because basically what is being used is the P. Okay, dia ada 3 P kan, ATP, GTP, triphosphate. Is the phosphate that is important. Sama ada guna satu fosfat ataupun dia guna dua fosfat menjadi pyrophosphate. Because sometimes the cell will also use 3 of the phosphate. Okay, so this is what is important, not the A or the G. Okay, sometimes the A and the G is also important, but in most cases what is important is the phosphate. Okay, jadi manusia GTP, plant animal is ATP, right? So, okay, so in ATP in plant bacteria, GTP in animal. Okay, basically no more reduction in carbon. Okay, at this point semua carbon jadi 4. Okay, 4 4 4 4 4. Okay, basically what happens here is either the addition of the phosphate or the taking of the phosphate or the rearrangement of the molecule structure or the production of some NADH or FADH2. Okay, jadi contoh inilah. So you have 4 carbon here. Okay, still 4 carbon. Okay, between succinyl and succinate. Okay, then from succinate, okay, cikgu itu kata saya suka succinate F fumarate. Okay, then okay, so between succinate and fumarate, okay, the enzyme that is involved is succinate dehydrogenase. Okay, basically again to take away another hydrogen. Okay, this is an oxidation that involves FAD. Okay, not NADH. Okay, FAD. Okay, so this is vitamin B2. Okay, so the mechanism involve hydrogen removal. Yalah sebab mudah because you use the enzyme dehydrogenase. Okay, it takes away hydride or the hydrogen atom. Okay, and it involves a deprotonation process also, right? So I'll just show you. Okay, so from succinate, okay, to produce fumarate. Okay, the number of carbon is the same. Okay, tapi apa yang berbeza? Yang berbeza adalah the removal of hydrogen. Okay, no more removal of carbon. Okay, so the 2 hydrogen is removed, one from here and one from here. Okay, and you get a double bond. Wait, sorry, double bond. Okay, so 2 ni pergi mana? 2 akan pergi NADH+. Okay, and, sorry, FAD+. Okay, kalau FAD dia akan ambil 2 hydrogen. Kalau NAD dia akan ambil 1 hydrogen. Right? So this is what happened. So please know the event and then know the enzyme. Okay, that is what is needed from you. And not only do you know the event, removal of hydrogen, removal of CO2, okay, but you have to know what is the subsequent metabolite. Macam kes inilah. This is double single bond jadi double bond. Okay, itu yang kamu harus hafal from this class, right? Okay, so from fumarate, M is malat. Okay, so from fumarate to malat. Okay, another enzyme that is used is called fumarase. Okay, so fumarase is the enzyme that makes this process possible. Okay, so basically hydration will occur across the double bond. Maksudnya air akan masuk. Okay, right? This is the trans addition of the element of water. Maksudnya air daripada persekitaran akan masuk. Right? Okay, so air akan masuk dalam fumarate. Okay. So basically, one of the OH will come here, one of the H will come here. So basically to break the double bond, right? So the OH is here, and then one of it will be here. Okay? So H2, dia akan ambil one HO, dia akan ambil satu H. Okay, they will park here. Okay, so please know the event. Okay, then this is mediated by the enzyme fumarase. Okay, dia memerlukan air untuk to produce malate from fumarate. Okay, and then just know that carbon is still the same. Okay, cuma they break the double bond, okay, to put in the OH and the H, right? So just now. Okay, so lastly, okay, is the production of malate. Okay, tadi kamu hasilkan malate, okay, malate to produce the final product, which is oxaloacetate. Okay, sama juga 4 carbon. Okay, so what happens here? Okay, what happens here is that more NADH is produced, but there is no more oxygen. There is no more carbon dioxide that is produced. Okay, it's just the taking away of one of the this one, the hydrogen. Okay, dia menjadi double O sini. Okay, double bond. Okay, so this H dia akan keluarkan bergabung dengan ni lah. Okay, not here actually, this one. It's this one. Okay, bergabung ni. Right? Okay, so basically that is what happens to the TCA cycle lah, right? So if you ask me, okay, what is the significance of the Krebs cycle? Okay, itulah the significant is basically to get rid of the carbon. Okay, sebab you started with 6 carbon tadi kan, the citrate. So you get rid of one, you get alpha ketoglutarate. You get rid of one more carbon, you get the succinyl CoA. Okay, so that is one importance. Okay, so basically the Krebs cycle, okay, the Krebs cycle is important in eliminating all the sugar that you makan. Okay, ingat lah, you makan starch, it becomes glucose. Glucose undergo glycolysis. Okay, so menghasilkan pyruvate. So the three carbon of pyruvate from your sugar tadi kan, tiga kan. So satu hilang during the formation of acetyl-CoA. Satu carbon hilang during the formation of alpha ketoglutarate. Satu lagi hilang dalam the formation of the succinyl-CoA. So basically all the sugar that you eat akan hilang dekat sini. Okay, all the 6 carbon you makan will be gone by the time the Krebs cycle enters the midway. Okay, so that's why some book or some biochemists, they say that the Krebs cycle is actually a wastage process. Okay, sebab itulah penat-penat you gather your carbon, okay, daripada makanan. Dekat sini dia sedap-sedap dikeluarkan dalam bentuk CO2. Okay, so some say this is a very a wasting pathway. Okay, but then again, ini kita perlukan lah. Kalau tidak, semua gula kita makan jadi gemuk lah. Semua jadi gemuk macam saya. Okay, right? But that is how lah. I mean, biochemically, this is how your sugar is being expelled. Okay, so dia bukan keluar daripada kentut, daripada tai kamu, daripada pedoh kamu, tapi dia keluar daripada Krebs cycle. Right? Okay. So the first thing is the reduction of the carbon number. Okay. Now the second important thing that is important in the TCA cycle is ATP production. Okay, ATP production either from the GTP or from the ATP or from the NADH. Kan you produce a lot of NADH tadi kan? You produce a lot of NADH to produce acetyl-CoA, to produce alpha ketoglutarate, to produce succinyl-CoA, and to produce oxaloacetate. So all these NADH will go into the next process, which is the oxidative phosphorylation. Okay, jadi itu yang kita akan masuk in the next kuliah next week lah. Okay, but basically ini yang berlaku lah, mengeluarkan carbon, menghasilkan ATP dan menghasilkan NADH. Alright? Okay. So by the end of the Krebs cycle, okay, this is what you produce. Okay, you started with acetyl-CoA tadi. Okay, the two carbon biomolecule. Okay, in the end you're going to produce 3 NADH. Okay, you're going to produce 1 FADH. Ingatlah, in between succinyl-CoA and fumarate. Okay, you produce FADH ni tadi. Okay, sometimes they letak QH2. Okay, some book that do this. Okay, but some book they letak FADH. Okay, so just know that this is FADH2. Okay, Q ni mewakili FAD. Okay, so you produce by the end of this process, you produce a lot NADH, you produce some FADH, you produce ATP or GTP for this case. Okay, and then you kehilangan 2 CO2. Okay, ingat, you kehilangan dekat mana tadi kan? Dekat the alpha ketoglutarate and the succinyl-CoA. Right, so this is the overall reaction for the Krebs cycle. Okay, right? Now satu lagi yang you have to know, mungkin ini ada diceritakan dulu in your first year. Now all this while when you learn about NADH and ATP production, selalunya dalam kuliah dia akan katakan satu NADH menghasilkan 3 ATP, betul kan? Lepas tu 2 FADH menghasilkan 2 ATP. Satu FADH2 menghasilkan 2 ATP. Now actually that is not so true. Okay, the real value is actually 1 NADH, okay, akan menghasilkan 2.5 ATP. Okay, and that should have been known to you in your first year dulu. Okay, I hope that you remember that. I think sebenarnya dah cover lah. So by the end of the ATP, okay, so what happened is that itulah, one NAD hash is actually 2.5 ATP. Okay, so um, one FADH, kalau kamu perayu dulu, kamu akan cakap 2 ATP kan, actually is only 1.5. Okay, jadi kalau dikira semua ni, by the end of the Krebs cycle, okay, 10 ATPs are produced, okay, actually from all this NADH and the GTP. Okay, the GTP ni ATP lah, right? Okay, so please take note of that, right? So this is again just the number lah. Okay, right? Now, in the Krebs cycle, okay, the Krebs cycle, it is also regulated. Okay, maksudnya Krebs cycle, they don't just happen by itself. Okay, they can be stopped or they can be pushed to go forward. Okay, maksud boleh di boleh di induce, okay, ataupun boleh di inhibited. Okay, so this slide just to show you there are some of the things that can inhibit. Okay, if you look at this red, yang merah, okay, yang red, this red thing. Okay, if you have too much NADH, okay, or too much acetyl-CoA, okay, this can become an inhibitor. Okay, while if too much NAD plus or coenzyme A, okay, or ADP or carbon, okay, this can induce that the cycle pergi ke depan, right? Okay, just to show you that one lah, daripada sini, right? So basically, when you look at the TCA, actually there ada 3 kalau tak 2. Sebenarnya ada 2 lah. It's actually at this one and this one. Okay, selalunya from pyruvate to acetyl-CoA, many biochemists they say, oh ini belum berlaku lagi. I mean the Krebs cycle have not happened yet, which is true lah, because the Krebs cycle happens here. Okay, during the addition of acetyl-CoA and also acetate. Only that some book they even include that pyruvate is a precursor for the happening of a Krebs cycle. Okay, so if you want to include pyruvate, there is 3 points of regulation. But if you want to be more strict and you say the Krebs cycle is upon the production of citrate, they actually 2 points. Okay, so you have to know what inhibits and what stimulates. Okay, calcium ion, they very much stimulate. Okay, sebab itu one of the thing kalau if you are not, or if you are lethargic, okay, again another thing that you can take is calcium pill. Okay, either calcium pill or you can even minum susu. Okay, so sometimes doctor will ask you to drink milk. Okay, yeah, milk has all sorts of other nutrients, but one of the things that milk can help you in metabolism when it comes to glycolysis or Krebs cycle. Okay, they have calcium. Okay, so it's the calcium that actually help with G production also, right? So if you have more calcium, the Krebs cycle becomes more efficient, right? Okay, jadi itu adalah TCA cycle, alright? Okay, so now so far what we talk about is that the Krebs cycle happens after the production of acetyl-CoA. Okay, so acetyl-CoA daripada mana tadi? Daripada pyruvate. Okay, now not only is the Krebs cycle happening when you have acetyl-CoA from pyruvate. Sebenarnya acetyl-CoA does not necessarily have to come from pyruvate. Okay, acetyl-CoA can come from other things. It can come from your fat. Okay, acetyl-CoA can even come from the protein that you eat. Okay, as long as there is an acetyl-CoA, dia tak kisah daripada mana. Okay, alright. So this next slide just shows that TCA cycle is also possible when you have acetyl-CoA from other means. Tak semestinya daripada pyruvate. Okay, now fatty acid and amino acid, they can also be broken. Okay, sama macam kalau you have glucose, you break it down into pyruvate. Okay, pyruvate break it down into acetyl-CoA. It's the same thing when you have fatty acid. Okay, fatty acid and amino acid, you can break it down. Break it down. In the end, you're still going to get the same thing, which is acetyl-CoA. Okay, so as long as there is acetyl-CoA from any sources, okay, from your glucose, from your meat, from protein, okay, it can feed, okay, it will feed into the Krebs cycle, okay, via the same process lah, glycolysis pun boleh, from whatever. Okay, so basically when you look at this one, okay, yang kita dah cover until now is that we look at the carbohydrate punya route, okay, daripada laluan karbohidrat, okay, which basically starts from glucose lah. Okay, so glucose, okay, all the way into pyruvate. Okay, so right, okay, so from pyruvate, then you get the 2 carbon of acetyl, okay, then the Krebs cycle. Okay, sebenarnya tak semestinya just from glucose. Actually, from protein, okay, if from protein, you have different types of amino acid. It can be also broken down. The amino acid can be broken down, okay, into keto acid. This keto acid can either go to produce pyruvate also. Okay, the keto acid can also produce untuk Krebs cycle. Okay, kita akan tengok nanti some example. Okay, and then also same thing for fat. Okay, so if you take fat, okay, it will still be able to give you glycerol, and this will enter the glyceraldehyde triphosphate, okay, which is yang GAP. Okay, remember last week kita bercerita tentang DHAP and GAP. So from fat you can get GAP, and from GAP we can get pyruvic acid lah, macam daripada glucose. So from fatty acid also, okay, you can even get acetyl-CoA. Okay, fatty acid, fat, you get the fatty acid. Okay, fatty acid yang 16 C carbon C16, C18, you break it down into, you potong, dia akan jadi 2. Okay, kalau dia jadi 2, dia akan jadi acetyl-CoA. Okay, so this same acetyl-CoA masuk sini lah. Okay, then become enter the Krebs cycle. Alright, so itulah, kalaupun kamu tidak makan, even if you don't eat rice, even if you don't makan mie, tak makan nasi, okay, you still gonna get acetyl-CoA from your protein. Okay, you still got, kalaupun tak makan protein, kamu minum lemak saja, okay, you still gonna produce acetyl-CoA. So okay, the acetyl-CoA. Jadi this acetyl-CoA again can be used to do other things lah. Right? Okay. So that is how your body works. Jangan kamu ingat, kalau diet jangan makan nasi, kalau diet jangan makan lemak. No, it all interconnects. Okay, because your body will scavenge the small molecule from the food that you eat. Right? Okay, this is just, okay. Kalau ini tadi, okay, this is one example from fat, okay, from carbohydrate, and from protein. Now this slide, okay, that I'm showing now is that, okay, these are the more specific example. Okay, contohnya from arginine, from glutamine, histidine, proline. Okay, these are all your amino acid. Okay, all these amino acid, this 4 group can produce glutamate. Okay, and glutamate will enter the Krebs cycle because glutamate can be converted into alpha ketoglutarate. Okay, sama juga isoleucine, methionine, threonine, and valine. When they are broken, okay, some part of the carbon akan menjadi succinyl-CoA. Okay, phenylalanine, tyrosine, if you eat them from your meat, okay, potong-potong, dia akan dapat menghasilkan fumarate. Okay, jadi maksudnya you get all of this not only from the initial acetyl-CoA tadi, the acetyl-CoA, okay, of course it will enter. Okay, but also from the meat that you eat, you will even produce all the different metabolite. Okay, jadi this is just to show lah. Okay, so macam ni lah, leucine, lysine, phenylalanine, tyrosine, okay, can either produce acetyl, then acetyl will produce acetyl, okay, then enter the cytosol, okay. Arginine, okay, the thyroxine from here, okay, can also be converted into fumarate. Okay, jadi banyak lah, like I say, whatever you eat, as long as there is C, there is O, there is H, okay, your body can somehow break them and turn them into acetyl-CoA. Right? Right? So selain daripada all the different amino acid that can produce all the different metabolite for citric acid cycle, okay, it can also be the other way around. Okay, selain daripada dia menerima benda lain, okay, all these metabolite can also be used and transported to produce other things. Okay, right? So what this slide, or what we're going next, is that the TCA cycle can also be the source of many other metabolite, because you have the citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, solicetate, dan semuanya kan, and the cotinase. You have all these metabolite. Okay, you can even use it for other reaction, not just for the Krebs cycle reaction. Okay, so the TCA cycle can provide intermediates for many other biosynthetic processes. Okay, jadi maksudnya tak semestinya citrate become isocitrate, tak semestinya isocitrate menghasilkan alpha ketoglutarate, Okay, sometimes when you have too much of the citrate, dia akan pergi tempat benda lain. And in fact, too much of citrate can even become fat. Okay, too much of the alpha ketoglutarate can become protein. Okay, jadi that is what it's trying to say lah. Meaning it's not just that intermediate, it's not just the metabolite becoming the next one. Okay, but if you have too much of one, dia boleh digunakan untuk buat benda lain. Okay, this is in your notes lah. Okay, so contohnya, okay, from the acetyl CoA. So acetyl CoA. Okay, this can become acetoacetyl. Okay, from acetyl CoA, this can even become steroid. Okay, acetyl CoA, okay, even become fatty acid. Jadi yang saya katakan lah, kalau kamu banyak sangat makan nasi ya, acetyl CoA, will become a fatty acid lah, right? Okay, alpha ketoglutarate, okay, can become glutamate. Okay, kalau tadi in the previous slide is the glutamate that becomes the alpha ketoglutarate. Okay, but if your citric acid cycle produces too much of alpha ketoglutarate, then not only can it become succinyl CoA, kalau banyak sangat dia pun boleh jadi glutamate. Okay, boleh jadi proline, boleh jadi anything else lah. So basically your body or our body is like a kilang lah. You produce all this metabolite either for Krebs cycle, or this metabolite can be used in other parts of other cell to produce other things, right? Okay, so just to show you lah, okay, that our body is actually very versatile lah, right? Okay, right now I'm going to go into the next part of the Krebs cycle, which is called the glyoxylate cycle. Okay, now remember when I was telling you about the some books or some authors, they say that the Krebs cycle is a waste. Okay, because you have all this carbon. Okay, masukkan menjadi citrate. From citrate becomes isocitrate. From isocitrate you lose one carbon dalam CO2. Okay, then after that you lose again the CO2. Mean it becomes shorter. Okay, so this is a wasteful cycle. Okay, now there is another cycle. Okay, that is called the glyoxylate cycle. Okay, the glyoxylate cycle is actually a Krebs cycle potong dua. Okay, it's half of the Krebs cycle. Okay, why? Because it doesn't want to the glyoxylate cycle, okay, is a modified version of the Krebs cycle, okay, whereby no carbon is released. Okay, no carbon is wasted. Okay, jadi maksudnya dia akan pergi dia akan jadi citrate, which is still 6 carbon. Dia akan menghasilkan isocitrate, which is still 6 carbon. Alright, but from isocitrate, okay, isocitrate dia tidak akan turun kepada alpha ketoglutarate. Why? Because you will lose carbon. Okay, you lose carbon. Okay, I think dia akan memang dia akan terus pergi succinyl CoA. Right? Now, the glyoxylate cycle, okay, is a version of the Krebs cycle. Okay, selalunya for plant and for bacteria. Okay, kalau plant selalunya di dalam biji, in seeds. Okay, biji kelapa sawit, biji jagung, biji getah, in the biji, right? Okay, so the reaction occurs in specialized organelle. Okay, these organelle are called glycosysome. Okay, kalau Krebs cycle tadi berlaku dari mana? Krebs cycle happens in the mitochondria. Okay, but the glyoxylate cycle doesn't happen in the mitochondria. It happens in the glycosysome. Okay, so this enable the plant to store carbon in seed as oil. Okay, jadi kita akan tengok nanti lah. So the glyoxylate cycle, okay, allows plant, okay, dia membenarkan tumbuhan menggunakan acetyl CoA. Samalah, you will still use acetyl CoA. Same process depan tadi, the production of citrate is the same. Okay, it use the acetyl CoA, okay, derived from the oxidation of fatty acid for carbohydrate synthesis. Okay, jadi animal cannot do this, okay, because our acetyl CoA is totally used dalam bentuk CO2. Jadi maksudnya animal can only undergo Krebs cycle. Okay, during the Krebs cycle we lose CO2, Okay, but in plant they can have a second, they can have another cycle, okay, which do not lose the CO2. Okay, and this cycle is called the glyoxylate cycle. Okay, I just show you ni lah. Okay, jadi now in the Krebs cycle, kita ada acetyl CoA, betul kan? Acetyl CoA bergabung dengan 4 carbon of oxaloacetate. Okay, then we produce citrate. Okay, so that is the same. Okay, now from citrate, in plant seed, okay, you will produce isocitrate. That's the same, okay, because at this point no carbon is being taken away, okay. But when you look at Krebs cycle, kan from isocitrate, the next metabolite is the alpha ketoglutarate, right? But when isocitrate is converted into alpha ketoglutarate, CO2 is produced, maksudnya dibuang. Okay, the carbon is terbuang lah, right? So this is what we don't want. Okay, this is what the seed, okay, don't want to do. Okay, so what happened is that instead of isocitrate dia menjadi alpha ketoglutarate. Okay, what happened is that isocitrate does not form alpha ketoglutarate. Okay, tapi apa yang berlaku adalah from isocitrate dia terus membentuk susinat. Okay, susinat is 4 karbon. Ingat tadi ya, 6, 6, 5, 4, 4, 4, 4. Okay, now from this case what happens is that isocitrate will produce or will go to susinat. Okay, jadi let us look at the Krebs cycle. So basically when you look at the glyoxylate cycle, okay, when you look at the glyoxylate cycle, what happens is that from citrate 6 carbon, isocitrate 6 carbon, okay, it doesn't go to the production of alpha ketoglutarate because this involves the losing of CO2. Okay, but instead in the glyoxylate cycle, isocitrate dia akan terus pergi kepada susinat. Okay, so this is 6 carbon, this is 4 carbon. So you ask yourself, 2 carbon lagi pergi mana? Kalau ini 4, kalau ini 6 carbon, tiba-tiba dia jadi 4 carbon. So 2 carbon pergi mana? So let's look back at the glyoxylate cycle. Now, so isocitrate 6 carbon. Okay, 6 carbon. So 4 of the carbon will directly go into the susinat. Okay, and this will go to fumarate, malat, oxaloacetate, biasalah. Cuma pada bahagian di mana CO2 itu dihilangkan in the alpha ketoglutarate, in the succinyl CoA, this does not occur. So what happens is that from isocitrate, 4 carbon pergi kepada susinat. Okay, then another 2 pergi mana? Then another 2 will go into the production of what is called glyoxylate. Okay, so itu nama dia, glyoxylate cycle, right? So glyoxylate is actually a 2 carbon molecule. Okay, 1, 2 carbon molecule. Okay, 4 carbon pergi dekat susinat and it undergoes the same citric acid cycle. Okay, but 2 will go into glyoxylate. Okay, so these 2 glyoxylate, okay, akan bergabung balik dengan extra acetyl CoA. Okay, so some of the acetyl CoA masuk melalui this one. Okay, and some of the acetyl CoA akan masuk melalui sini. Okay, so acetyl CoA 2 carbon. Okay, glyoxylate 2 carbon. Okay, so 2 tambah 2, dia dapat 4. Okay, so dia akan masuk malat. Okay, malat is 4. 1, 2, 3, 4. Okay, so at this point, what happened is that no carbon dioxide is produced. Okay, basically in the seed, okay, dalam biji benih, what happened is that they try to conserve so that no carbon is dihilangkan. Okay, so the carbon will maintain in the biji benih, okay, dalam bentuk glyoxylate. Okay, 2 carbon again, okay, tapi dia akan bergabung dengan acetyl CoA untuk membentuk 4 carbon of malate. Okay, so the malate will gabung lagi untuk membentuk apa? Dia akan membentuk glucose. Okay, jadi malate will gabung dengan dengan oxaloacetate, dengan acetyl CoA again, lah. Okay, so it bypass this one to produce 4. Then this 4 will gabung lagi dengan oxaloacetate, dengan acetyl CoA, which is another 2. Okay, dia akan jadi 6. And this is 6 is actually glucose. So from here, glucose is produced again. So when you produce more glucose, buat glucose, one glucose, then this glucose will start to bercantum. Okay, so what happened? This glucose will start to bercantum, dia akan jadi cellulose. Okay, that is why when you tanam biji benih, okay, kalau kamu tanam biji benih, kamu herankan macam mana dia boleh menghasilkan akar. Okay, macam mana dia boleh menghasilkan pucuk daun. Okay, because those are cell wall. Betul kan? Akar and the pucuk daun is like cell wall. And what are cell wall? Cell wall are cellulose. And what are cellulose? Cellulose are long chain of glucose, macam starch. Okay, starch is also long chain of glucose. Cellulose is also long chain of glucose. Okay, jadi carbon is not released. but it is gabung untuk membentuk malet. Malet akan bergabung balik dengan acetyl-CoA untuk membentuk glukos. Okay, dan gabung balik glukos, dia akan menjadi cell wall. Okay, then start jadi akar. Sebab itu biji benih, okay, dia start boleh jadi ada akar, ada tu. Okay, jadi memang keajaiban lah. Okay, so daripada mana akar dia datang? Daripada ni lah. It's because of the acetyl-CoA. That's why biji benih selalunya banyak lemak. Okay, you have minyak kelapa sawit, minyak jagung, minyak kuaci, the sunflower seed, rapeseed oil, minyak, banyak-banyak lagi minyak lah. Okay, why? Because this minyak, okay, will actually be converted into acetyl-CoA. So from acetyl-CoA membentuk glyoxylate, glyoxylate membentuk malet, campur balik dengan acetyl-CoA, which is from the oil. Okay, so membentuk gula lah. Gula membentuk apa? Membentuk membentuk apa nama tadi? The cellulose. Okay, so you see. So, jadi next time when people say, kenapa tanam jagung boleh keluar pucuk? Kenapa tanam benih boleh keluar pucuk, boleh keluar akar? It's because of the fat that is converted not into CO2 but converted into back in the glyoxylate membentuk gula balik. Right? Okay, now there is a question just now. Tunggu, saya cari dulu soalan tu. Someone asked me a very long question. Kejap, let me find that question first. Chat. Yeah. Okay, someone asked me in the chat. Banyak soalan chat ni. High surface area kan doktor? Itu saya tak faham. Kalau nak dapat secara naturally macam mana doktor? Nak dapat apa ni naturally? Vitamin. Kalau nak dapat vitamin naturally from food lah. Okay? From food. Okay? Because we are end user. Okay, kalau kamu nak dapat vitamin daripada naturally, then you have to makan sayur lah. Okay, because sayur atau tumbuhan, they are synthesizer. Okay, they have the ability to synthesize cellulose, to synthesize vitamin and macromolecule because of the sun energy. Okay, kita tak boleh. Kita have to eat them to get energy. So, jadi kalau nak dapat naturally, kena banyak makan sayur lah. Banyak makan sayur, banyak makan tumbuhan lah. Okay, because they produce that one. Okay, apa lagi soalan ni? Since I ambil biochem, ada ambil multivitamin. Kudos. Okay, doctor, in glyoxylate cycle, the fumarate, okay, will become malate. Okay, where does the other two carbon go? Mana? Tak ada. Apart from the malate, okay, dia punya soalan tadi kan. Okay, from the fumarate, dia akan menghasilkan malate. Okay, jadi, okay, this malate, okay, if you see this one, okay, this malate is produced from the fumarate. Okay. Now this malate can also be produced by dua carbon. Ini dua carbon dua carbon. So you get ni. So you get malate. Meaning in glyoxylate cycle you will produce a lot of malate. Okay. Kalau in the normal Krebs cycle, one cycle will produce maybe one malate. Okay. Now in the glyoxylate cycle you will still produce malate. Okay. Memang ini pun produce malate from fumarate. But you produce also more malate because acetyl-CoA from fat, okay, and the glyoxylate will produce more malate. Jadi maksudnya more malate lah, dia more malate. So what happens to the excess malate? Now the excess malate, if I'm not mistaken, dia akan bergabung balik dengan acetyl-CoA, meaning there are more acetyl-CoA lah from the fat. Okay, so malate is 4 carbon, acetyl-CoA 2 carbon, dia akan jadi ini 2 kan, jadi 6. So what is this 6? This 6 is glucose lah. So this glucose gabung dia akan jadi cell wall. Okay, I hope that answer your question right? Okay, right, thank you. Alright, okay, jadi ni balik. Okay, so this is what happened in plant seed. Okay, in plant seed they also have mitochondria. Okay, they also have glyoxysome. Okay, is that specialized organelle. Okay, so this glyoxysome tak ada pada haiwan, tak ada pada manusia. Okay, we have mitochondria. And when you have mitochondria itulah you need to take pyruvate and acetyl-CoA daripada gula lah. Alright. So what happened with seed is that in the mitochondria, okay, they still undergo the Krebs cycle. Okay. Only that when they have in the glyoxylum, okay, what happens is that when in the glyoxylum, in the glyosome, okay, the citrate, okay, this is where the normal Krebs cycle happens in the mitochondria. But in the gliosome, okay, the citrate go to produce isocitrate 6 carbon. Okay, this 6 carbon, okay, dia akan menghasilkan succinate. Okay, with succinate can either be used back in the gliosome, or succinate will be transported into mitochondria. Okay, then it will undergo the Krebs cycle. Or the isocitrate will produce, dia akan menghasilkan glyosylate yang akan balik dengan gabung balik dengan acetyl-CoA from fat. Okay, menghasilkan malat. Okay, malat akan bergabung, okay, untuk membentuk isoroacetate. Then at one point dia tak tunjuk sini lah, dia tunjuk a few arrow ni, maksudnya dia banyak lah, there's a few reaction. Okay, usually dia akan bergabung balik dengan some acetyl-CoA lah. Okay, then they will produce glucose. Okay, jadi when you have enough glucose, dia akan gabung, then they become the cell wall lah. Right, so this is just another showing of the Zelet cycle. Right, and we're done. Okay, right, any question or not? Okay, so what we cover today is on the TCA cycle. Okay, so TCA cycle start with the pyruvate daripada luar. Okay, from daripada luar sel. Okay, sorry, daripada luar mitochondria. Then it's transported into the mitochondria matrix. Okay, where it undergoes process by TPC tadi. Okay, the TPC will convert the 3 carbon pyruvate into 2 carbon acetyl-CoA. Then the 2 acetyl-CoA gabung dengan 4 oxaloacetate, jadi dia jadi 6 citrate. Then you start lah all those processes. And in between every intermediate, you have to know the enzyme. You have to know what happens to the metabolite, right? So then it's a cycle again. Okay, so basically what happens, okay, is the production of ATP, production of FADH2, okay, production of NADH. We produce a lot of NADH because that is what you want to produce ATP. So what again happen? It's the reduction of carbon number. Okay, daripada 6, 6, 5, 4. Okay, so where all this carbon goes to, keluar menjadi CO2. Right, so that is the TCA cycle. Okay, then a second version of the TCA cycle is the gliosylate cycle, which is actually half of it. Okay, kalau TCA cycle, the carbon that you get dibuang, in the gliosylate cycle, the carbon is conserved. Okay, untuk membentuk lebih malat. Okay, so you produce more malat, eventually you produce back glucose. Okay, the seed we use it to produce pucuk lah, untuk produce akar. Okay, untuk produce dia punya pucuk. Alright, boleh ah? Okay, right. So I hope you understand that, or I hope this is something new for you lah. Right. So we're going to stop for 10 minutes. Okay, then we're going to come back. Then we'll take attendance. Right. Okay. So.